about DNA, we think about it in terms of genes and being able to code for proteins and traits. That is largely termed gene expression. So when you hear the phrase gene expression, you should think, how are we going to show what that DNA is coding for? This is usually thought of as the central dogma. So it can be called the central dogma of molecular biology, of genetics, or it's just central dogma in general. But it's talking about the same thing. How do we get the message out of DNA and into a protein? So central dogma largely is saying that DNA can make more copies of itself through replication, that DNA can be transcribed into RNA, and that RNA, which is an mRNA molecule, can be translated into proteins. So transcription is the process by which we go from DNA to RNA. Translation is the process by which we go from mRNA to a protein. So we're going to first turn our attention to transcription. So I told you what it is. Where does it occur? It's going to occur wherever the DNA is located. So in eukaryotes, this will be the nucleus. In prokaryotes, this will be the nucleoid. And it occurs as needed. So whenever you need to make the functional product, be it a protein or an RNA, transcription will occur. So those two broad classes of our genes, you have protein coding genes and non-protein coding genes. So if it's a protein coding gene, this is the traditional gene that we think of, and it's going to be transcribed into mRNA, and that mRNA is going to be translated into a protein, and that protein is going to be modified, or the polypeptide will be modified to make a functional protein product. If it makes an RNA molecule, then that DNA will be transcribed into that RNA, and that RNA will be edited to make a functional product. Some examples are rRNA and tRNA. Long non-coding RNAs exist. So rRNA makes ribosomes. So ribosomes are made out of RNA and then some proteins in it. tRNA is made out of RNA that has been modified. But the tRNA and the rRNA are the functional products. So back to transcription, here's our DNA molecule. You have regulatory sequences. That's a binding site for regulatory proteins that increase or decrease the rate of transcription. So you can add an enhancer or a silencer, and they're going to either increase transcription or decrease that transcription. The promoter is a binding site for polymerase, RNA polymerase, and this is going to be largely where transcription starts. And then you have a terminator, which is a sequence that says, hey, stop. That's the end of this gene. Now, when we make that mRNA molecule, so you see it starts with the promoter, and it ends around the time where the terminator is. It has a ribosome binding site, a start codon, the codons that are in between, and then a stop. This is what's necessary for translation. So the DNA has regulatory sites for transcription, and the RNA has regulatory sites for translation. Like with replication and the same with translation, transcription can be broken down into initiation, elongation, and termination. So during initiation, polymerase needs to bind to the promoter. During elongation, polymerase is going to go along, opening up that DNA, letting it close behind it as it transcribes the message. So the message is going to be coming out of the polymerase. So polymerase is opening it up and making it, and as it elongates, or as elongation continues and we get further along the DNA, we get a longer and longer molecule of RNA until we hit termination, when everything falls apart and our DNA is back the way it was. Our polymerase leaves it alone, and we have an RNA transcript that can leave the nucleus and go on to make a protein. Some other things to know. There's a non-template strand and a template strand. We talked about the promoter. When you hear upstream, it's usually towards the 5' end, and downstream is towards the 3' end. But upstream is talking about anything that's upstream of this transcription start site. This transcription start site is usually denoted with a +1. Okay? +1. Now anything that comes before, anything upstream, is going to be denoted with a negative, -35, -5. Anything downstream, a positive, +100. And now we're talking about the location of different sequences. Okay. Most of these sequences are consensus sequences. So these are like sequences that have common base pair order. So they can vary on some notation. So you'll see some like, oh, here's a T, here's an A, here can be any pyrimidine, here can be any purine, here can be anything, here can be a C or a G. So they are consensus, meaning, you know, a lot of the letters in that area is the same, but it can differ. So back to the template strand. The template strand is the strand that's going to be used to transcribe. The template strand is also called the antisense strand. The non-template strand is the sense strand. You will notice that the non-template strand and the RNA molecule that's being made have the same direction, and they also have the same sequence with the exception of T's and U's, okay, because it is an rRNA molecule, but essentially they are very similar. It's also important to note that either strand of DNA, okay, so remember DNA is a double-stranded molecule. Either one of those strands can be used to transcribe. The genes on either strand can be used for transcription to make an RNA, but if you use it on the negative strand here, you won't use it on the positive strand, okay? So even though you can use either one, each gene knows which one it should come off of, but you can't do both. So here, A and B are on one, C is on the other, but you're not going to say, oh, A, B, B, C, C, one or the other for transcription. So this is just a basic overview of transcription. These are some things that you're going to find common in bacteria and eukaryotes, but we're going to go into more detail in both of those.